start now. Okay, class, so we're continuing on our genetics unit, and we already talked about, what have we talked about in the past? We talked about mitosis, meiosis, DNA, so we have an understanding of that. And then last class, we also talked about Mendel. And who is Mendel? He is the father of genetics. Yes. So what we're going to do today is we're going to just review the 3 laws of Mendel's laws of heredity. We're going to learn some new terms in genetics, and we're going to do a method, actually we're going to explore, called Punnett squares. And we're going to talk about that more a little bit later. Okay? So, first off, some review from last class. So the Mendelian laws of heredity, there's 3. So can we have, Katie, can you read the first one?
Okay, so segregation. Alleles are separated into individual gametes to transmit genetic info to offspring during meiosis.
Yeah. So what word jumps out at us there?
Alleles.
So, do we remember what alleles are? It is the alternate forms of a genetic trait, right, in a chromosome. So what this, all it says is that during meiosis, that alleles separate, and then one goes into each gamete. Okay? So Dave, can you read the dominance one?
The dominant allele completely masks the effects of a recessive allele, and thus always expressed.
Okay, so we understand we have dominant and recessive. So do you remember what the difference is between them?
There's 2 alleles, and depending on if one's dominant, one's recessive, the other one will take over the other and express it.
So that means we'll see the dominant, not the recessive. And then, so the independent assortment, can you read that law again, Katie?
Yes. Alleles on different chromosomes are distributed randomly to individual gametes.
Okay, so that just means that some genes may be passed on while others may not. It's a random. Okay? So, now, do we have any questions we remember from last class?
We're good.
All right, we're good. So, some new terms in genetics now. We're going to look here. So, we have a genotype. So, Dave, can you read what a genotype is?
Genotype are the alleles an organism contains for a particular trait. For example, capital T, little
you don't
t.
have to say it. Yeah, so we can have different types of genotypes. So genotype means what the actual gene looks like with the alleles. So each part, so we have the big T and the small t, those are both alleles. Okay?
Yep.
So now a phenotype. Katie, can you read that?
Yes. The observable trait based on interaction between genes and the environment.
Yeah. So an example is our height or eye color. So these are the observable. That's the main point we're going to highlight right there, okay? That's kind of confusing with genotype and phenotype, but essentially genotype is different types of genes we may have, but the phenotype is what we see, and we'll talk more about that once we do Punnett squares. So also, we may see these words pop up a lot, and we have homozygous and heterozygous. So have we heard the beginning part, homo, before? Where we've heard that before? That is appropriate.
The same.
Homologous.
It means the same.
Yeah, homologous. The same, right?
Yep.
So even homosexual, that means a male and a male may like each other, or a female and a female may like each other. They have the same sex, right?
Yep.
And then heterozygous. So the main part here we're highlighting is hetero. So that means I have different, right? So that means a male and a female, almost like in heterosexual. They both like each other.
Yeah.
So if we use it in terms of alleles, we have homozygous, so we'll have like 2 big T's or 2 small T's. Or in heterozygous, what would we have then?
Big T and small t.
Like big T and a small t. Okay. Do you have any questions with our terms so far?
Nope.
Nope, we understand?
Yep.
Okay. So now we're just going to flip this over. Start our next activity. All right. So, what our main activity today is going through Punnett squares and the method. What we're going to use, though, is the monohybrid cross. So that involves one allele pair. So we're just observing the possibility of one trait. So we're going to be looking at eye color. So for the Punnett square method, it's actually a chart, and what it shows is the possible combinations that we may have passed down from a parent to an offspring. Okay, so what our activity is, so we're going to wait before we start drawing. I know we're trying to get ahead of ourselves. We're gonna do an activity with practice. So we're going to draw and complete a Punnett square, which is what this is. We're going to go through it together. And then we're going to analyze the table, and then we're going to look at these characteristics. So what does this kind of look like? It's really simple, the Punnett square.
A window.
Yeah, almost like a window. So what we could do, an easy way, is a window with two little ticks. Or sometimes I like to do is almost like another way is like a tic-tac-toe, right? So it can look like this almost. And then we just don't use this, and we use these boxes here. Right? It's kind of simple. All right. So we're going to use different colors because we like colors. So in order to have a child, we need to have two parents, right? For an offspring. So we're going to use both as parent 1, so P1 and P2. So let's say for eye color, we have one, we're going to use, let me use this for correction. Use big B as brown, okay? Brown eye color. And we're going to use little b as blue. All right? So, we're going to say that one parent has brown eyes, but it's a heterozygous. So that means, what does hetero mean?
Different.
Different. So that would be, what would that look like? A big B and a small b. Okay? So the second parent has blue eyes, but it's homozygous trait. So what would that be? Katie, what do you think?
2 small b's.
2 small b's. Okay. So what we're going to place now is one parent up here. So we're going to put one b there and the small b there. And then we're going to put the other parent on the side. Okay? Do we have this so far? So now we're going to complete the chart. So what we're going to do is just fill it in. It's very simple. We're going to put big B, and then a small b, and then we're going to put across B, 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 B. Okay. So, based on genotype, we know genotype is the different types of alleles that we may have. What are the similar ones here? What's similar between the 4? Which ones are the same?
2 left and 2 right.
Yeah, so this and this, right? So what we have a B, B, and then we have a small b, right? And how many times do we see it each? We see 2. Twice. So, if we have a big B represents the dominant one. That is the dominant gene. So, we have the big B means brown, and the small b means blue. So together, what do the big B with the small b represent? Brown eyes, right? So the 2 phenotypes we have is brown, 1, and then what would the other one be? Blue. Close. Right? Because then we have small and small. So 2 small make blue. So do we understand how the different genotypes versus phenotypes now?
Yep.
Mm-hmm.
Okay? And then we can count as well how many homozygous traits do we see and how many heterozygous traits you see.
See 2 homozygous and 2 heterozygous.
Exactly. So 2 and 2. So now, we just distinguished right there the number of genotypes and phenotypes. And we distinguish which are homozygous and which are heterozygous. The last thing we're going to do, what Punnett squares does, it allows us to see the possible combinations of Punnett square, right? So what does that also sound like? We use that word more in math, but we can also use it here.
Probability
and ratios, okay? So what is the probability then of a child either having brown eyes or blue eyes?
2 and
2
2.
and 2. So that's where we would add. So 2 versus 2. Do we have any questions about that?
Nope.
No? Okay. So in our textbook, there is a bunch of questions that describe different scenarios with simple monohybrid cross problems. And so we're going to practice doing our Punnett squares then. We're going to do a little bit in class, and then we're going to come back and take up some of them. Okay?
Yeah.
All right, so we're going to start with that. But first, before we do that, why would you think it is important to learn and solve basic genetic problems? What are we trying to do with the Punnett squares?
Guess the outcome of
traits, right?
Yes.
So that's what we're trying to do. Why do we think this may be of an interest to certain types of, like, different contexts or different professions? Think about it. If we can manipulate traits into something, in offspring.
GMOs.
So we hear that word a lot. So that has to do with farmer crops. So we will discuss these more later on, but I just want to keep that in mind with everybody as we look more into problems, genetic problems. Okay? So we're good for now. We're going to do the activity. We're going to do our practice questions, and then we're going to take it up. Okay. All done.